study how seagrasses control methane emissions from coastal marine environments. Methane is a very potent greenhouse gas, and its concentration in the atmosphere continues to increase and thereby enhances global warming. Most of this methane is of biological origin, meaning it is produced by microorganisms that use very simple organic compounds to form methane. Methanogenesis represents the terminal step in the degradation of organic material, and common substrates that fuel methanogenesis include hydrogen, acetate, as well as small methylated compounds such as methylamine or methylated sulfates. And the microorganisms that perform methanogenesis are strictly anaerobic archaea, the so-called methanogens, that use these compounds and liberate methane as the end product. And so in the environment, methanogenesis typically occurs in anoxic areas where abundant organic material is present, and this includes, for example, marine sediments. And marine sediments are really hotspots for methanogenesis. However, if we look at methane emissions from marine environments, we see that although much methane is produced in marine sediments, only relatively little is eventually emitted to the atmosphere. And this is because these methane emissions are strongly mitigated also by microbial activity, so by a microbial process that is called methane oxidation, that very efficiently removes methane from the environment. And therefore, the microorganisms that perform methane oxidation are considered a very important biological methane filter. The interplay between these two processes, so between methane oxidation and methanogenesis, ultimately determines how much methane is emitted from the environment. It is currently assumed that about half of the global methane emissions are originating from aquatic ecosystems, and this includes freshwater environments as well as marine environments. By far, most methane is emitted from freshwater environments, including lakes and wetlands, whereas the emissions from the marine environment are typically relatively small. But while relatively little methane is emitted from the open oceans, the vast majority of methane emissions actually originates from coastal marine environments, including shallow coastal areas that are vegetated by seagrasses. So in our research, we investigated the mechanisms behind microbial methane production and consumption in sediments that are covered by Posidonia oceanica seagrasses in the Mediterranean Sea. Seagrasses are in general very well recognized to play an important role in the marine carbon cycle. They are highly productive ecosystems, so they fix and sequester large amounts of carbon dioxide from the atmosphere and store it inside their sediments, and therefore they are highly valued as important blue carbon ecosystems. And the capacity to store carbon in their sediments is particularly pronounced for seagrasses belonging to Posidonia oceanica. So underneath these seagrass meadows, the carbon deposit can extend up to several meters in thickness in sediment. However, seagrasses have also been shown to be sources of methane, and methane is the much more powerful greenhouse gas in comparison to carbon dioxide, and these methane emissions therefore do partially offset the blue carbon function of these ecosystems. The processes that ultimately lead to methane production inside sediments that are covered by seagrasses are so far unknown. So in our research, we basically wanted to understand the mechanisms behind the methane production in sediments that are covered by Posidonia oceanica seagrasses, where we specifically looked at the metabolic processes that occur in these sediments that lead to methane production or consumption, what controls these metabolic processes, and how this eventually contributes to methane emissions from these ecosystems. To answer these questions, we used a combination of different methods. We performed flux measurements to quantify how much methane is produced and released from the sediments into the water column. We performed substrate addition experiments where we basically tested for the utilization of different methanogenic compounds. For this, we collected sediment cores from underneath a seagrass meadow and tested for the utilization of the typical methanogenic substrates, including hydrogen, acetate, as well as methylamine, and also for the utilization of methane for the process of methane oxidation. And we also performed chemical analysis in order to identify potential methane precursor molecules that are present inside the actual environment. The combination of these different methods then allowed us to get a comprehensive understanding of the metabolic processes and on their controls in the sediments which we investigated. Our experiments showed that sediments covered by Posidonia seagrasses were indeed a net source of methane. And most of this methane was produced in the surface sediment layer, and the rates then tended to decrease with depths. At the same time, we could not detect substantial rates of methane oxidation, suggesting that the biological methane filter in this environment was not very active. We further found that in these sediments, methane was only produced from one particular type of compounds, namely the methylated compounds, including methylamine and methylated sulfides, but not from acetate or hydrogen. And this was intriguing because acetate and hydrogen represent the typical methanogenic compounds in marine environments, whereas methylated compounds are generally considered to not contribute substantially to methane emissions from marine sediments. When we then looked for the source of these methanogenic compounds, we found that the methylated compounds were actually abundantly present in the tissue of the seagrass plant, suggesting that the methanogenic activity was actually coupled to the seagrass plant and that the plant itself was the source of these methanogenic substrates in this environment. We then wanted to go a step further and investigate how the death of the seagrass plant would actually affect methane production in this environment. For this, we investigated a site where the living seagrass plant had died off more than 20 years ago, so meaning that the superficial cover of the seagrass meadow has vanished, but where the sediments still contained the dead plant material. What we found was that basically the methane production rate in the sediment were highly similar to the methane production rates that we found underneath the living seagrass meadows. And interestingly, the dead seagrass tissue did still contain the methylated methane precursor molecules, implying that these compounds could actually persist in the tissue for several years after the death of the plant, suggesting that the methanogenic activity in these sediments can be retained for decades after the seagrass plants have died. So our study provides a very comprehensive understanding on how methane cycling is controlled in these seagrass ecosystems, and this will help us to better assess the role of these ecosystems in the global methane cycling. Our study highlights yet another negative consequence that the loss of seagrass meadows has on our climate. Because once the seagrass meadow dies, on the one hand, it loses its capacity to sequester carbon dioxide from the atmosphere and therefore loses its blue carbon function. But at the same time, these ecosystems continue to release methane. And this is a very important aspect which we have to consider in our efforts in restoring these important ecosystems. So far, we have focused our studies on sediments that are covered with Posidonia oceanica seagrasses in the Mediterranean Sea. But globally, there is actually a large diversity of seagrass species, which all vary in their sizes and also in the amount of carbon that they deposit in their sediments. So it would be an interesting next step to investigate if the same mechanisms underlie methane production also in sediments that are covered by other seagrass species, or if perhaps other factors may control methane production in these ecosystems.